I'm John Mu, a research engineer here at Bina Technologies. Today I'll be introducing our gold standard genome for validating structural variants. There is great need in the community for a gold standard set for validating structural variants. There are many structural variant calling algorithms out there, such as MetaSV, Lumpy, Deli, and Pindel. However, almost all of them are validated with only simulated data or small real data validation sets. Today we'll introduce a large real data validation set for structural variants. To generate this real data validation set, we leverage some existing data sources, such as the reads from Craig Venter's genome. These were long Sanger reads of up to 1,000 base pairs. They also included many back clones with large insert sizes, useful for validating insertions. We further generated a high coverage dataset of high-seq reads, and all of this data has been released on SRA. Here I'll briefly go over the construction procedure we use to generate this gold set. We use the method of multiple forms of validation. The initial set of variants comes from the original paying set from the Venter genome. We also augment this set with additional variants that we call from BWA and BLAT. This set of variants then go through multiple forms of validation, including junction mapping, paired end mapping, and concordance with DGV. Variants which pass two forms of validation are included in our gold set. Since this gold set is not comprehensive, it can only be used to estimate the true positive rate. In order to estimate false positive rate, we construct regions called no structural variant regions. These are basically regions which have high coverage of Sanger reads with very low edit distance to the genome and high mapping quality. If an SV is called in one of these regions, it is likely a false positive. These regions cover about 60% of the genome. To put these two things together, we can compute both true positive rate and false discovery rate. Given a variant that was called by a variant caller, we first check whether it exists in the gold set. If it does, then it's a true positive. Otherwise, we check if both breakpoints are in the NSVRs. If both are, then it's a false positive. Otherwise, it's excluded from analysis. This estimate of true positive rate and false discovery rate are useful for the comparison of tools. Since if one tool has a higher true positive rate and lower false discovery rate compared to another tool, it is likely to also compare favorably to the other tool in the whole genome, since these regions represent relatively easier regions in the genome. This plot shows the number of variants in our gold set divided into size bins, and also compares against some other gold set of variants that exist in the community. BGS represents the Baylor gold set from the Baylor College of Medicine. GIAB is the Genome in a Bottle sets, and Platinum is the Illumina Platinum Genome set. Our gold set compares favorably in terms of number of variants to all of these other sets. Finally, we use our gold set to compare some popular structural variant detection algorithms. All algorithms perform favorably in terms of true positive rate, detecting many of the SVs in the gold set. However, BreakSeq2 and MetaSV stand out as having quite low false discovery rate. In conclusion, we have presented a large and comprehensive set of structural variants for use as a gold set in comparing structural variant callers. We showed that this gold set was useful in determining which structural variant callers were more accurate. We hope you'll also use our gold set in comparing your own structural variant callers and provide us any feedback.